Recording problems. Yeah. So, welcome back. Sorry for the technical glitch. So, we'll continue. Shall we continue? Yeah, that's fine.
Yes, sir. Yes, sir.
Yes, wonderful. So, let me begin with, let's say, hemostasis. So, I think this is what we discussed in the beginning. So my current talk is focused on the last component, characterization of molecules of therapeutic importance from venoms. Particularly, the toxins interfere in hemostasis. So as a result, this is the title, as I already discussed. So let me begin with hemostasis. It means a tightly controlled, a tightly regulated, or highly structured crosstalk between plasma coagulation and platelet aggregation process in innate immunity. Innate immunity means the immunity that is derived from the birth. So thus, basically, hemostasis is an acute phase inflammatory response to tissue injury in innate immunity. So now let me define snake venom. Snake venom is a complex, very, very complex mixture of predominantly enzymatic and non-enzymatic protein and peptide toxins. Interestingly and incredibly, most of the toxins are target specific. They do target vital organs, vital systems such as nervous, muscular, and circulatory systems. These three systems are tightly interconnected, interlinked, such that effect on one system invariably affect the other two, resulting in immobilization or killing of the prey animal or human victims. Thus, snake bite is a true, very true example of a simultaneous lethal assault on vital organs or vital systems. Thus, it's a true medical emergency. So this is the SDS page pattern of the saw-scaled viper venom. You can see, this is the molecular weight marker, and these are the toxin bands resolved in SDS page under non-reduced condition. You can see many bands have been resolved here. So it's indicating a complex mixture of proteins. Actually, SDS page is a less sensitive, somewhat less sensitive technique. And, its sensitivity is very less. I think you require at least minimum 3 to 5 microgram to visualize the protein band. But if you analyze this same venom in sophisticated high-end technologies like RP-HPLC or ESIMS or HPLC-MS techniques, you can see over 100 different peaks, suggesting that snake venom is really a complex mixture of enzymatic and non-enzymatic protein and peptide toxins. See, Glenn King from Australia is working extensively on Australian spider venom, the funnel web spider venom. He has characterized, he has mentioned the presence of over 1,000 protein toxins in spider venom. So indicating that venom is truly a mixture of protein and peptide toxins. So thus many venoms and their toxins directly affect the circulatory systems, so particularly hemostatic system. So as I already mentioned, hemostatic system means it involves platelet aggregation and plasma coagulation events. If you take platelet aggregation, many of the toxins may induce aggregation. That means there will be formation of the platelet plug, and many of the toxins may inhibit the aggregation of platelets. So this will platelet plug formation or no platelet aggregation as an impact, a strong impact on the conditions like hemorrhage and all. And in case of plasma coagulation, many of the toxins may induce coagulation. Those toxins or venoms that induce coagulation are popularly called procoagulants in nature. And many toxins or the venoms may inhibit coagulation process, and such toxins or venoms are called anticoagulants. So thus, the venom toxins interfere in platelet aggregation and plasma coagulation. They may induce or inhibit. Thus, they interfere in hemostasis. Now, let me give you the gist of hemostasis. Hemostasis, as I already mentioned, is an acute phase response to a tissue injury. It is characterized by platelet activation, or it involves platelet activation, clot formation, and clot dissolution. Whenever there is a vascular injury, blood vessel injury, immediately platelets get activated. They adhere to the damaged site. Right on top, there will be formation of clot, which seals of the damaged site. It is something like platelets act like bricks over which we have the clot, the cement. A temporary wall is created. Thus, the minor hemorrhages that are taking place, the day-to-day events, are easily taken care of through this mechanism. If we take the blood coagulation cascade, blood coagulation process, we have different pathways, as you are aware of. Like we have intrinsic pathway, it is also called contact activation pathway, and extrinsic pathway, it's also called tissue factor pathway. These two pathways are activated through different mechanisms that I think I have highlighted here. And despite the fact that they're initiated through different mechanisms, they culminate at one point, they join at one point. From this point onwards, we have a common pathway. So the common pathway starts from the formation of factor 10A, that is tenase complex. The tenase complex is a complex of factor 10A, factor 5A, and calcium, and also membrane phospholipid. Membrane phospholipid is contributed by platelets. So activated platelets contribute membrane phospholipids. So we have tenase complex. So from this point onwards, we have the common pathway. So in this case, finally, the prothrombin is converted into thrombin. Thrombin cleave its substrate fibrinogen and convert fibrinogen into fibrin. This is what the clot, is it not? This is a fibrin monomer, is the soft clot. Eventually, this fibrin monomer then cross-linked through factor 13A, and it becomes a hard clot. So it's a strong, clot. So, so we have intrinsic pathway and extrinsic pathway, we have common pathway. And the interesting factor associated with these pathways is that each factor involved in any of these pathways are activated through limited proteolysis. That is, each factor is activating the other factor through limited proteolysis. If you want to know what limited proteolysis means, you can ask me a little later. And the other important property that is associated, you can clearly notice, is the property of fibrinogen. Fibrinogen is a large molecular weight structure, but once it is subjected for limited proteolysis, it forms fibrin. So if you look at the properties of fibrinogen and fibrin, fibrinogen is soluble while fibrin is insoluble. See, just the limited proteolysis will convert the soluble fibrinogen into insoluble fibrin clot. So here, the efficiency of the final clot formation obviously depends on the efficient functioning of all the clotting factors. So if there is an impairment in any of the clotting factors, results in impaired clot formation, resulting in bleeding disorders. In bleeding disorders, the factors, any of the factors does not perform properly, normally, then you don't end up in the formation of right clot. So it leads to bleeding disorders. And I also already mentioned many of the toxins strongly interfere in coagulation process. So the toxins may interfere in intrinsic pathway, toxins may interfere in extrinsic pathway, toxins may interfere in common pathway. So common pathway involves the tenase complex formation only and thrombin generation. And later, the fibrinogen is independent. So fibrinogen and we have fibrin. So toxins may even act on fibrinogen and as well as fibrin and interfere in the process of clot formation. So this cartoon is showing the development of platelets. Platelets are enucleate or anucleate tiny cells which are biconvex, discoid in shape. This is the scanning electron micrograph of platelets. platelets are derived from the common hematopoietic stem cells of the bone marrow. So hematopoietic stem cells of the bone marrow, depending on the requirement, so we have all blood cells originate from the common pluripotent stem cells. Depending on the requirement, they take up a particular lineage. If the system needs lymphocytes, then this hematopoietic stem cells will convert into lymphoid lineage, and we have lymphocytes are formed. If you take the lymphocytes, we have basically T lymphocytes, B lymphocytes, and natural killer cells. So those are involved in immune responses. Let us don't worry about that. And rest of the blood cells, except lymphocytes, rest of the blood cells are derived from myeloid lineage. So in myeloid lineage, again, depending on the requirement, whether the system needs platelets, RBCs, or monocytes, or polymorphonuclear leukocytes. So these myeloid lineage cells take up a particular route. So in this case, in myeloid lineage, if the system needs platelets, then the myeloid stem cells convert into megakaryoblast, which later converted into megakaryocyte. Megakaryocyte is a multinucleate giant cell. You can see over here, we have many nuclei inside. So it is derived due to fusion of several cells. We have megakaryocyte. So it's a multinucleate giant cells. From these multinucleate giant cells, megakaryocytes, platelets are pinched off structures. So which are the enucleate structures. So platelets are otherwise called thrombocytes. We know that platelets are made in the bone marrow. They are derived from the bone marrow. And all these years, we have the opinion that the platelets are made exclusively in the bone marrow. However, recent studies indicated the fact that even the platelets are formed in the lungs. See, Verich and Zimmerman in 2013 had got the idea of the platelet generation in lungs. And in 2019, by this group, Looney and Lefrançais, they discovered using intravital microscopy technique that the platelets do form in the lungs. So platelets are generated from the lungs through megakaryocytes only. So the way megakaryocytes gives platelets in the bone marrow, in a similar way, megakaryocytes sits in the lungs, interstitial space of lungs, give rise to platelets. So nearly 50% of the platelets that are made every day are even derived from the lungs. Thus, this is the recent discovery that platelets are made both in the bone marrow as well as in lungs. And the important thing is that how exactly these multinucleate giant cell megakaryocytes travel from bone marrow and sit in the lungs and give rise to platelets. That part is not clearly understood. Okay. So this is what. And now let me give you the gist of platelet aggregation. As I already mentioned, platelets are enucleate, tiny cells. So this is the scanning electron micrograph. So in the resting state, platelets appear biconvex, the discoid like structure. So this is in the resting state. When the platelets are activated, they change their shape like this. These are the activated platelets. See here, when platelets are activated, you see a lot many changes over here. So activated platelets eventually undergo or exercise degranulation because platelets store lot many granules inside them. So the granules are of three types over here: alpha granules, dense granules, and lysosomes. Alpha granules contain over 20 to 30 different components. So I've listed a couple of them here. I've just given the subtitle ligands in the alpha granules: fibrinogen, wild membrane factor, fibronectin, vitronectin, thrombospondin, P-selectin, and so many components are stored in alpha granules. And dense granules, over 40-50 components are stored in the dense granules. In dense granules, we have polyphosphates, very critical: ATP, magnesium, ADP, GDP, serotonin, chemokines, factor 13, alpha-2 macroglobulin, platelet factor 4. Likewise, we have about 30 or 40 different components are stored in the dense granules. And take into account the lysosome, you are already aware of the fact that lysosome is a suicide bag of the cell because it has lot many hydrolytic enzymes. You name any substrate, you have enzyme to degrade in lysosomes. That is the speciality of lysosome. So if you take a platelet molecule, platelet cell, a platelet cell will have about 50 to 60 alpha granules, about 8 to 15 dense granules, about 3 to 5 lysosomes. The number of lysosomes present in platelets are very less, and the number of alpha granules so far exceedingly high. So activated platelets undergo degranulation, first thing, and then they shape change. They undergo shape change like this. So in this condition, they become sticky. Once they become sticky, means they tend to aggregate each other. See, this is what I have represented in the cartoon here. Let's say this is the platelet in the resting state. See, look at this. This is circular shape, round, and most important, whether platelet is activated through any agonist, the end result is the same. So finally, what happens is the platelet activation take place, shape change take place like this, and during this process, the receptor, that is fibrinogen receptor, it's glycoprotein 2b3a complex, or it is also called alpha 2b beta 3. This complex becomes active. See, in the resting platelets, this complex, its conformation is such that it cannot bind its ligand fibrinogen. Let's say I mentioned in the cartoon here, this is a fibrinogen, it cannot bind fibrinogen. But once it is activated, platelets undergo shape change, and the receptor also undergo conformational change. Now, the conformationally altered receptor now can bind the ligand. So here, see, look at here, this fibrinogen red things, dumbbell-shaped structure I showed this is fibrinogen. Even Von-Willebrand factor. Von-Willebrand factor will cross-link the platelets. The major cross-linker is the fibrinogen. So cross-linking take place like this. So resulting in platelet aggregation. See here, the thing is that resting platelet, we have the shape change and the change in the conformation of the fibrinogen receptor or Von-Willebrand factor receptor, and the agonists are plenty here. Shear stress can be an agonist. It can activate a platelet. Epinephrine can activate platelet. Collagen can activate platelet. Thrombin can activate ADP, thromboxanate, arachidonic acid, platelet activating factor, ristocetin, antigen antibody complex, calcium ionophore. Many of them activate platelets and cause aggregation. And snake venom toxins. Venom toxins, many of the venom toxins also activate platelets. So the activators, you can simply call them as agonists. So these agonists can stimulate platelets and cause aggregation. Okay. So see, these aggregation, the binding is facilitated through RGD or KGD sequences. See, fibrinogen has RGD sequence. RGD sequence is much essential to bind to the fibrinogen receptor. This is most important because RGD or KGD sequences have been explored in designing antiplatelet drugs. Okay, that's what I'm trying to mention here. So this is the gist of platelets. Now, as I said, whenever there is endothelial damage, invariably you see activation of platelets. When platelets are activated, invariably you see the activation of blood coagulation. These are connected. So these are all the important responses, preliminary or acute phase responses in inflammatory response. You take inflammation invariably, triggered on due to damage, tissue damage or vascular damage. So damage, endothelial damage. So endothelial damage exposes the collagen subendothelial layer, and as a result, platelets bind to exposed collagen in the subendothelial layer. Platelets aggregate, platelets activate. On the activated platelets, you have the blood coagulation. The activated platelets, as I mentioned here, see, platelet activation results in degranulation. Degranulation results in the release of so many things. Ligands, several ligands are released. See, these are all ligands are released. Several components are released. See, polyphosphates are released. Polyphosphates, I don't know whether you have heard of or not. Polyphosphates is simply a phosphate, inorganic phosphate linked through anhydride bond. So it's a chain of phosphate. polyphosphates are formed. So in one chain, there could be 50 to 100 phosphates linked and forms a polyphosphate and gives a negative charge, and that helps in actually the activation of the blood coagulation. In the intrinsic pathway is activated basically, and that needs a negative charge for the activation. And this is where the polyphosphates actually contribute for that. And in addition, platelets, the activated platelets, exhibit phosphatidylserine on the outer surface. Phosphatidylserine, impart, charge on that. And that also gives charge for the activation of plasma components right on the surface of platelet. But polyphosphates are the predominant ones. Because of polyphosphates, intrinsic pathway is getting activated. Okay. See, and also ruptured cells. When there is a damage over there, cells will rupture. As a result, at that local milieu, there is a drop in pH. Drop in pH you can come across. So the drop in pH, reduction in pH, is responsible for Hagemann factor 12, and prekallikrein, macroglobulin, polyphosphate, this complex to get activated. They form a complex. In that Hagemann factor, autoactivation takes place over here due to reduction in pH and formation of complex. So autoactivation factor 12A is formed. So 12A is responsible for, is a starting component for the intrinsic pathway, coagulation event. And in addition, factor 12A also activate prekallikrein to kallikrein. So we have kallikrein system. Kallikrein system is a system of serine proteases that you remember probably. And this kallikrein act on kininogen. We have kininogen, like see here, macroglobulin is a kininogen. So kallikrein act on kininogen and gives rise to kinins. The most popular kinin is a bradykinin. It's not. Bradykinin, you are aware of the fact that bradykinin is a strong vasodilator. I think 1 microgram of bradykinin can enlarge your blood vessel 6 times its original size. That means, supposing if the blood vessel is occupying about 1 ml means, 1 ml blood means, if you add 1 microgram of bradykinin, now it expands in such a way that it can accept 6 ml of the blood. So much of vasodilation take place. So bradykinin is known for that. Bradykinin is a strong vasodilator. Okay, thus, whenever there is vascular damage, endothelial damage, immediately invariably platelets get activated. When platelets get activated, invariably blood coagulation is triggered on. So these are the responses. They're acute phase responses in inflammatory event. So these are part of inflammatory response in textbooks, and people generally do not consider this together while discussing inflammatory response. Actually, inflammatory response, these are the starting component. Whenever there is a damage, that's eventually inflammatory response is triggered on. This is critical. You have to overlap. You have to inculcate these things. So as I already mentioned, snake venom is a complex of so many enzymes and non-enzymatic toxins. See, phospholipase A2, so phospholipase B, phospholipase C. You can see proteases. We have serine proteases, metalloproteases. We have collagenases, elastases, fibrinogenases, kininogenases. These are all proteases. Acetylcholine esterases degrade acetylcholine, heparinases degrade heparin, hyaluronidases degrade hyaluronic acid, amino acid oxidase act on amino acids, deoxyribonucleases act on the respective substrate ribonucleases, ATP hydrolases, AMP hydrolases, pyrophosphatases. We have series of enzymes. Even you have many more enzymes that I'm not listing these things. And so thus venom is mixture of several enzymes, enzymatic toxins. These are all not simply enzymes. In addition to enzymatic activity, they exhibit toxic properties. They involve in toxicity. That is most important. And we have non-enzymatic toxins also. We have 3-finger toxins. Basically, 3-finger toxins are the neurotoxins. We have both presynaptic neurotoxins as well as postsynaptic neurotoxins. You can come across disintegrants. Disintegrants, as I already mentioned, they contain RGD sequence. So disintegrants, they can inhibit the clot formation. So platelet aggregation. They can inhibit platelet aggregation at places of fibrinogen binding. These small structures, they can go and bind and inhibit fibrinogen binding. As a result, platelet aggregation may be inhibited. So disintegrin will have RGD sequence or KGD sequence. RGD means arginine, glycine, and D-glutamic acid. D-aspartic acid. That is the sequence. KGD means lysine, glycine, and D-aspartic acid. That is a sequence. Is very much essential. There's CTLs means cytolysins are cardiotoxins. Snake legs means C-type lectin like structures and protease inhibitors which are non-enzymatic. BPPs means bradykinin potentiating peptides and nerve growth factors and natriuretic peptides. So that natriuretic peptides regulate the blood pressure. So in addition, there could be several non-enzymatic toxins. I am not listing all of them. These are some of the well-characterized, well-known non-enzymatic toxins. These are the well-known enzymatic toxins. And I'm happy to tell you the fact that the toxic role of hyaluronidases and deoxyribonucleases, our laboratory established that. Before that, people thought that hyaluronidases and deoxyribonucleases, they just involved in digestion of the ingested prey, not involved in killing, that is not involved in toxicity. But our lab has demonstrated, has discovered the fact that hyaluronidases and deoxyribonucleases, they play a very important role in making the venom less toxic or more toxic. This is what hyaluronidase, its properties has been established long ago, during 2003 or 4 from our laboratory, from my group. And deoxyribonuclease, as I mentioned, we published a paper in Nature Communication in the beginning while working with local toxicity of viper venoms. In that, we have demonstrated the role of deoxyribonuclease, the toxic role of deoxyribonuclease. Okay, so these are the various pharmacological properties exhibited by snake venom poison: edema, that's swelling; hemorrhage, oozing of blood; neurotoxicity, maybe presynaptic or postsynaptic; myotoxicity; cardiotoxicity. Myotoxicity may be working on smooth muscle or skeletal muscle; cardiotoxicity working on cardiac muscle; and hemolytic, lyse the RBCs; coagulant, effect on blood coagulation; platelet aggregation; hypotensive, reduce the blood pressure; hypertensive, increase the blood pressure; methemoglobinemia and hypoxia; netosis or etosis. Methemoglobinemia and hypoxia, netosis and etosis, these properties, our lab established that. Our lab discovered these two. Okay, so coagulant and platelet aggregation, as I already mentioned, coagulant, there could be procoagulants or anticoagulants. Platelet aggregation, they may induce aggregation or they may inhibit aggregation. And coagulation, plasma coagulation, platelet aggregation, they are the heart and soul of the hemostasis. So this is just to give you the summary of protease, which we have the properties, this summary of the properties of the protease NNPF3, which we have purified from Indian spectacled cobra naja venom. You can see over here, this is the SDS page pattern. This is the C line, lane C. You can see over here, single band. And so those days, during 1997 or 98, we purified this. That time, showing single band in SDS page itself is a big thing. So we demonstrated that in SDS page. And eventually that time MALDI-TOF was introduced. MALDI-TOF also, we used MALDI-TOF technology to characterize its molecular mass. So MALDI-TOF mass spectrometry revealed its molecular weight as 67.82 kilodalton. And the enzyme activity, it's a protease. It's basically protease isolated from this venom. And this protease, its activity was inhibited by EDTA and EGTA. You know that EDTA is a divalent metal ion chelator. It's a non-specific one. EGTA is specific for calcium ion, is it not? So EGTA can chelate calcium ions. EDTA is a non-specific one. It can chelate any divalent metal ion. So our enzyme is inhibited by EDTA as well as EGTA. And the atomic absorption spectrometry confirmed the presence of both zinc and calcium in the molecule. So thus our protease is a metalloprotease. And don't worry about that. So this protease belongs to P3 class of snake venom metalloproteases. P1, P2, P3 classes of snake venom metalloproteases. There is classification. Don't worry about that. So our enzyme belongs to P3 class of snake venom metalloproteases. And when we checked for the toxic properties like edema, hemorrhage, neurotoxicity, cardiotoxicity, myotoxicity, cytotoxicity, lethality, our enzyme was devoid of all these toxic properties. So that means our enzyme was found to be non-toxic, no toxicity. However, whatever concentration you purify and inject into an organism, it is not going to harm the organism. It is non-toxic. No induction of any of these toxic properties. However, our enzyme strongly interfered in hemostasis. Particularly, it exhibited strong anticoagulant activity. That is, inhibited clot formation, and it's also exhibited strong platelet aggregation inhibition. So it inhibited platelet aggregation. Thus, our protease strongly interfere in hemostasis. So thus it's showing anti-hemostatic effect. It is inhibiting strongly both plasma coagulation as well as inhibit aggregation of platelets. So these properties are very, very essential and can be explored for the human benefit in different forms. Now, the interesting thing is that having understood that our protease is a strong anti-hemostatic protease, then we wanted to understand like how does the protease exhibits anti-hemostatic effect. So hemostasis means, as I already mentioned, it involves plasma coagulation and platelet aggregation, is it not? So does our protease interfere in these two functions? So to begin with, we targeted initially anticoagulant activity, that is inhibition of plasma coagulation process. So here we tested the plasma recalcification time. So you can see over here, our normal plasma would clot around 130, 140 seconds. But in presence of our enzyme, you can see the clotting time increased, showing the strong anticoagulant property. So this is the dose-dependent effect. With increasing doses of our enzyme, we can see the clotting time also increased. And this is the time-dependent effect. See, we have, so we took the plasma and pre-incubated with our enzyme. We used 1 microgram of our enzyme and incubated with different lengths of time, 5 minutes, 10 minutes, 15 minutes, 20 minutes, 35. Like this, we incubated different lengths of time. So as the time progresses, we can see the plasma coagulation is inhibited. Thus, our protease exhibited strong anticoagulant activity. It is inhibited plasma coagulation process. Then the interesting thing is, what is the mechanism of anticoagulant activity? That's an interesting one. So for biochemists generally, if you have something, next level would be understanding its molecular mechanism is the general interest. So as a result, we asked a question, what is the molecular mechanism of anticoagulant activity? 1 minute, I'll switch off my phone. Sorry, it's in the on mode. Yeah, I put it up. Let me drink a little water. Okay. So the next thing for us, the interesting thing was, what is the molecular mechanism of anticoagulant activity exhibited by our purified protease? For this, we checked the various parameters, like we targeted intrinsic pathway, extrinsic pathway, common pathway. So through which pathway our protease actually works. So we checked APTT, it activated partial thromboplastin time. This APTT means activated partial thromboplastin time, PT, prothrombin time, TCT, thrombin clotting time. So to our surprise, we found that in all the cases, we observed anticoagulant activity. So our protease, suggesting that our protease is not working through intrinsic pathway or extrinsic pathway or common pathway. If it is not working through these three pathways, then where exactly it works? The remaining two things in the pathway are the fibrinogen, conversion of fibrinogen to fibrin, our protease would may be acting on fibrinogen and depleting the potential clotable fibrinogen, therefore no clot formation. Or our protease might cleaving the formed clot. If it cleaves the formed clot, there also you don't see coagulation, is it not? This is what. So thus, our protease, we see the delay in clotting time. Then we ask the question, is the anticoagulant activity due to degradation of fibrinogen or degradation of fibrin clot or degradation of both of them? In either way, in either cases, you see no clot formation. Since our protease is not working through intrinsic pathway, extrinsic pathway, or common pathway, remaining thing only these two. Fibrinogen, our protease might be degrading fibrinogen, eliminating the clotable fibrinogen, or it might degrade the formed clot. As a result, we observe the anticoagulant activity. So we wanted to test that. So in that, we targeted fibrinogen degradation. See here, look at this. This is just plasma protein. We have, displayed, analyzed through STS page in non-reducing condition. So you can see over here, this is the plasma protein. So you can see over here. This is, you can see the arrow mark here. Can you make out arrow mark here? Can you see the arrow mark? Yeah, that's fine. So this one is actually a fibrinogen band. So our plasma protein was treated with the increasing concentration of our protease. Increasing concentration. See, look at this. This is the plasma here, and this is the loading control. We have used fibrinogen in a native loading control. Okay, you can see over here our enzyme. This is the control. No enzyme here, 0, and with increasing concentration of our protease, you can see specifically fibrinogen is depleted. That band is vanished. So that means our protease is targeting only fibrinogen and no other plasma proteins. Look at the specificity over here. This is very, very crucial in case if you want to explore this protease for the next level. So our protease is highly specific for the fibrinogen only. So this is the plasma protease, the plasma protein analysis. And this is with the purified fibrinogen. So purified fibrinogen, look at this here. So this is the control purified fibrinogen. So fibrinogen has A alpha, B beta, and gamma chains. We have, they all, assemble in dimers. 2 A alpha chain, 2 B beta chain, and 2 gamma chain, they form a complex. So if you take fibrinogen, in the non-reducing condition, if you analyze, you get a single band like this. If you analyze in a reduced STS page, you can see 3 bands, A alpha, B beta, and gamma chain like this. So this is with no enzyme. This is the control fibrinogen. This is with increasing concentration of our enzyme. You can see over here, 0.51, 1.52, 2.5 micrograms of enzyme was incubated with the fibrinogen here. So as the concentration increases, our protease is degrading A alpha, B beta chain. A alpha chain and B beta chains are vanished here. Look at here. Is it not? You can see clearly. This is the dose dependent effect. This is again time dependent effect. So in time dependent effect, A alpha chain vanishing is clearly observed. B beta chain. So let's say if you observe under densitogram, you can see the cleavage here also. So, and this is with again with different things. This is control. This is our enzyme. It is degraded, extensively incubated here, degraded both A alpha and B beta chain. And this is 110 phenanthroline. 110 phenanthroline is a metalloprotease inhibitor, but however, in this case, it is not inhibiting due to some other reason. So here also activity, you can see EDTA is completely inhibiting the activity here. So it is as good as the control. EGTA is also inhibiting, and PMSF is not inhibiting. PMSF, phenyl methyl sulfonyl fluoride is a serine protease inhibitor. Even one lupeptin. Lupeptin is again, it's an inhibitor for the cysteine and serine protease. So this is also not inhibiting this. So these are the molecular weight markers. So thus our protease is degrading specifically the fibrinogen. A alpha chain is preferentially degraded over B beta chain, while gamma chain remained resistant. This is what the result we got. Okay, you can see over here clearly, A alpha chains are degraded in preference over B beta chain. Okay, between these two, A alpha chain is preferentially cleaved, and then cleaves B beta chain, then gamma chain remain resistant. So now, anticoagulant activity is due to the depletion of clotable fibrinogen. This is one thing we have established. So this we got it published elsewhere. See, you have you have seen, like in COVID condition, people talk a lot about D-dimer. D-dimer, remember? See, if you take, this is the cartoon showing the fibrinogen structure. I said it is a 6 subunit structure. It has 2 A alpha chain, 2 B beta chain, and 2 gamma chain. They all aggregate and forms the fibrinogen, this complex structure. So it forms 3 domains: E domain, central domain. It's what we call head. D domain is called tail. This is another D domain tail. We have 2 tails and 1 head. So E domain is a head. So blue, D domains, 2 tails here, D domains, is it not? See, when fibrinogen is subjected to limited proteolysis, it becomes fibrin. See, limited proteolysis, these peptides, A peptide and B peptide are cleaved. So, okay, it's gone. Then it becomes fibrin monomer. This fibrin monomer becomes a soft clot, and this soft clot aggregate in head to tail fashion. Is it not? It forms a 3-dimensional network of fibrin clot. So that's why fibrin clot is formed like this. See, in COVID patients, people extensively monitor the D-dimer. See, D-dimer is actually a clot degradation product. Clot degradation product. See, look at this. This part has a D domain and see E domain and 2 D domains here. See, look at this tail D domain. So this is what we call D-dimer. People use it to monitor D domain to assess the clot degradation in COVID situation. Okay. So thus now anticoagulant activity is definitely due to degradation of fibrinogen, clotable fibrinogen. And anticoagulant activity could also be due to depletion of fibrin clot, as I already mentioned. If fibrin clot is degraded, that also responsible for anticoagulant activity. In order to understand, we did the fibrinolytic activity of our protease. This is the fibrin control. We have alpha polymer, gamma dimer, alpha chain, and beta chain. This is the control. With increasing concentration of our protease, you can see alpha polymer is vanished. Alpha polymer is cleaved and alpha chain is also cleaved. That means it is degrading, our protease is degrading the fibrin clot. This is a dose-dependent effect, and this is the time-dependent effect. And the degradation property is inhibited by EDTA. Look at this. This is in presence of EDTA. These two are EDTA treated ones. So EDTA is inhibiting the fibrinolytic activity of our enzyme, suggesting once again our protease is a metal protease. So alpha polymer and alpha chains were effectively degraded. We used washed plasma clot for this study. And this is again to show the fibrinolytic activity in a different method. This is through SDS page under non-reduced condition. And this is just a plate method over here. So what we did was we made the plasma to clot along with the agar. The plasma plus molten agar, calcium chloride is used to initiate clot formation. Is it not? We kept it for 12 hours. We have the gel formation over here. And we used different doses of our protease with increasing concentration of our protease. See, A is 1 microgram, 2 microgram, 3 microgram, and 4 microgram. E is actually PMSF. PMSF is actually a serine protease inhibitor. In presence of serine protease inhibitor, no inhibition is safe. You can see, our protease is degrading the clot. You can see the translucent background here. Is it not? Otherwise, rest all it's opaque. This is translucent. Degradation, so translucent regions indicate the degradation. And F, EDTA. EDTA is completely inhibiting. G is the buffer control. U, urokinase. Urokinase, we kept it as positive control over here. What urokinase does is it activates the plasminogen. Plasminogen is activated to plasmin. Plasmin eventually degrade the clot. See, the real degrader of the clot in vivo is the plasmin. So urokinase does that job. Urokinase activate plasminogen and convert plasminogen into plasmin, and plasmin would degrade the fibrinogen. So this is what you can see over here. And we prepared the whole blood clot as well as plasma clot, and we tested the degradation. See, this is the whole blood clot degradation profile, and this is plasma clot degradation profile. So here we can see over here, both plasma clot and blood clot are degraded. But the thing is that plasma clot is efficiently degraded over blood, but it is degrading both. It's most important. See, that means, see, in vivo, it has to degrade the whole blood clot. In case if it's a potential, drug candidate, this is most important. Our protease is degrading the blood clot as well. It is anticoagulant. It is degrading the blood clot. Most important. So thus, the anticoagulant activity of our protease is due to degradation of fibrinogen, that is fibrinogenolytic activity, as well as fibrinolytic activity. I mentioned both of them in one sentence. Fibrinogenolytic activity. It includes both fibrinogenolytic activity and fibrinolytic activity. So thus our protease is inhibiting coagulation, degrading both clotable fibrinogen and fibrin clot as well. Oh, so sir is already there with me. He's saying time is up. 5 more minutes he has given. And anti-hemostatic effect. See, so far I discussed the anticoagulant activity in terms of inhibition of plasma coagulation, inhibition of clotting of plasma, is it not? And the anticoagulant activity could also be due to inhibition of platelet aggregation. This is most important. As I already mentioned over here, platelets in the unstimulated state, they are like this. When they are stimulated, they undergo shape change. Lot many things happen. They undergo degranulation, they shape change, and they become sticky, and eventually they aggregate. Is it not? The most important thing in this case is we have agonist. If any agonist stimulate platelets, platelets get activated, platelets undergo shape change like this. And the most important thing is that it's a glycoprotein 2B3A complex fibrinogen receptor. It changes its conformation. So in this conformation, it cannot bind the fibrinogen. See, fibrinogen, how the fibrinogen is there in the blood, as well as it is released from alpha granules. See, wherever platelet activation take place, alpha granule degranulation take place, alpha granules contain a lot of fibrinogen. You have to remember. So those fibrinogen molecules are utilized in forming the, in cross-linking the platelets, resulting in aggregation of platelets. So see, I mentioned about disintegrins, remember? See, disintegrins. See, KGD or RGD sequence, remember? Let's say, see, this dumbbell, this shape, structure is important for binding the receptor, platelet receptor. It's a cross-linking. Supposing if you use the blue one, look at this. Let's say this blue one can replace this circular dumbbell shape. Is it not? See, now in presence of drugs which target platelet aggregation inhibition through fibrinogen receptor, let's say disintegrins, so many things, agrastat, integralin, eptifiphstat, absimax, monoclonal antibody, these are all, they target this alpha 2b beta 3 receptor, that's fibrinogen receptor. So in presence of these drugs, there will be inhibition of aggregation of platelets. They will not allow the fibrinogen to intercalate, bind. So hence there is inhibition of platelet aggregation. So this is what it is in general. So now we tested the platelet aggregation inhibition by our protease. So we tested in presence of several agonists. We have used thrombin, arachidonic acid, distocytin, epinephrine, ADP, and collagen. So you can see over here, thrombin, arachidonic acid, distocytin did not inhibit did not inhibit the platelet aggregation of our protease. So okay, so epinephrine induced aggregation and ADP induced aggregation was partially inhibited, while collagen induced aggregation was completely inhibited by our protease. You can see over here, this is 100% aggregation in the absence of our protease. In presence of our protease, there is inhibition of aggregation. So there is 100% inhibition. Collagen induced ADP and epinephrine, there is partial inhibition, while here no inhibition. Okay. So this is the collagen induced aggregation trace. So aggregation we just followed using turbidimetry. See, this is, you can see over here, this is the collagen alone, induced aggregation. So turbidity, turbid means the solution becomes thick, is it not? If turbidity is decreased, it will pass more, large light, is it not? Most of the light is passed through. So that means this is collagen induced aggregation. In presence of our protease, the dose dependent effect, you can see the inhibition of aggregation here. So on 150 nanomolar concentration, complete inhibition was achieved. So you can see over here. See, it has an IC50 value of 75 nanomolar. So our protease is inhibiting collagen induced aggregation at nanomolar concentration. This is most important, nanomolar concentration. And see, we tested our protease, the EDTA treated protease as well as native protease without inhibiting with any agent. See, EDTA, if you treat our protease with EDTA, EDTA will knock off the enzymatic activity. Is it not? So even if you knock off the enzymatic activity, so our protease is showing, platelet aggregation inhibition. And this is the native protease. Native protease is inhibiting the aggregation. Thus, both, EDTA treated as well as native protease inhibit platelet aggregation. So that means to say that it indicates clearly that our enzyme inhibits platelet aggregation through two mechanisms. One is enzymatic mechanism as well as non-enzymatic mechanism. So non-enzymatic mechanism, we have, I mentioned in the beginning, P3 class of protease, snake venom protease, P3 class of snake venom protease, they have a disintegrant domain. Disintegrant domain has RGD or KGD sequences, is it not? So our protease belongs to P3 class, it has a disintegrant domain, so hence it inhibits platelet aggregation. So, and it inhibits platelet aggregation non-enzymatically through disintegrant-like domain, and it inhibits platelet aggregation through enzymatic activity also. That enzymatic activity inhibition, that requires other story. This is what I think. I am short of time. This is a very interesting one. Anyway, had I given another maybe 20-30 minutes, I would have explained the different mechanisms over here beautifully. See, these are so many things there. I would like to summarize. We worked at the receptor level. See, conclusion. I would like to give you a conclusion for this. See, how does collagen inhibit platelet aggregation or induce, not inhibit, induce platelet aggregation? What collagen does is, collagen has two platelet receptors, alpha 2b, alpha 2 beta 1. This is one receptor, and glycoprotein 6 is this another receptor. These two receptors are collagen receptors. Alpha 2 beta 1 is a minor receptor, and glycoprotein 6 is a major receptor for collagen. Okay, so when collagen binds, it sends a signal transaction where a lot of tyrosine kinases are involved. Okay, tyrosine kinases, SARC family, LCK, tyrosine kinases are involved. Those are important in the degranulation. Collagen act, there is a degranulation. The granules undergo degranulation, release granular structures. Let's say fibrinogen. Fibrinogen is available for aggregation. Promotes degranulation, and also this activate the fibrinogen receptor. Fibrinogen receptor in the unstimulated state, it's like this. It cannot bind fibrinogen, but when it's stimulated, it changes its conformation. Now it can bind. Is it not? So collagen does this. Collagen does two things through signal transduction. That is, it causes degranulation and it causes the change in conformation of fibrinogen receptor. So finally aggregation takes place. What our protease does is, in this case, our protease binds to the minor collagen receptor, alpha 2 beta 1, and then it cleaves the major collagen receptor, glycoprotein 6. This is what the mechanism we established finally. Okay, it cleaves. As a result, it inhibits signal transduction. Inhibits signal transduction. As a result, platelet receptor, that is fibrinogen receptor, is not activated, and no degranulation. As a result, no platelet aggregation. See, this signal transduction we have demonstrated here. Look at this here. Look at this here. See, this is the washed platelets you have used. We have stimulated with collagen here. Look at this. With collagen, we have stimulated. We can see lot many, intracellular proteins. They have phosphorylated and tyrosine residues. SIC, SARC, LAT. These are all SARC family tyrosine kinases. Non-receptor cytosolic tyrosine kinases. They are very essential for the signal transduction. They get activated like this, right? This is very essential. This is what happens here when collagen stimulates. The signal transduction take place. So we have degranulation, we have receptor activation take place. But in presence of our protease, what happens is, look at this, in presence of our protease, the tyrosine phosphorylation rate decreases. Look at this, this we have demonstrated, okay? So it inhibits signal transduction. So this, eventually, we have demonstrated the binding of our protease to alpha-2 beta-1 and cleaving glycoprotein 6. So our protease prevent the collagen binding and also cleave the collagen receptor. As a result, it exhibits anticoagulant activity or inhibits platelet aggregation. That's what we have demonstrated. In summary, thus we have isolated and characterized a non-toxic and non-lethal metalloprotease. Metalloprotease inhibited blood clotting, removes clottable fibrinogen, dissolves whole blood and as well as fibrin clots, inhibits platelet aggregation, it prevents collagen binding, masks alpha-2 beta-1 integrin, and cleaves glycoprotein 6. As a result, it's an important candidate to explore as a drug candidate in order to treat the CVDs or stroke. You can use this conveniently to treat CVD and stroke after proper further study. This is my group. See, these are all. See, actually, this work was initially done by Dr. D.K. Jagdisha. Is currently is based in US, and this was further continued by Dr. Kumar. This is the work done by these two people, Dr. D.K. Jagdisha and Kumar, and these people are lab people. We have all assisted for that. And this is Dr. Girish. He's a professor. He also, he was initially worked with me as a doctoral student. He always with me. And we always cater the needs of our students, PhD students. So these are all my PhD students. And all these things is because of my PhD students. They have done all these things. So I thank them wholeheartedly. And my acknowledgements to Professor T. Virapasapagowda, with whom I did my PhD degree in University of Mysore. Before joining University of Mysore, I was a PhD student at Indian Institute of Science, worked with Professor P.S. Sastry. P.S. Sastry in biochemistry, he initially introduced me for the research work. I really thank these two great people wholeheartedly. And my senior in Indian Institute of Science in Sastry's lab, Professor Ram Rajshekaran, he was also ex-director of CFTRI. He always very kind to me. And my senior, Professor B.S. Vishwanath, is always, highly encouraging. So I thank all these people, funding from different agencies. And we used anti-alpha-2 beta-1 antibodies. Those anti-alpha-2 beta-1 antibodies, way back in 2003 or 4, we requested Professor Barry Kohler, University, USA. He kindly sent this antibody as a gift. So I really thank him on this occasion. Thank you very much for your patient listening. So in case if you have questions, you can ask me. You can ask
me.
I? Yes, sir.
Sir, thank you, sir. That was a great presentation. We came to know about a lot about the venom and how it impacts. So I would like to have this basic question. You said that there is a great lot of venom variability due to geographical location.
Yeah.
Is it so that if there is my first question would be like, if there is a change in the size of the venom sac, and secondly, I would like to know whether based on the size of the prey, these venom variability happens in different geographical locations, because when we take, when compared to the forest and cities, the prey is different for a particular snake.
Exactly.
Obviously, the venom sac also changes in like architecture or morphology, like whatever it is, and also the venom variability. You mean that this is the venom variability that happens, or to the ingredients actually?
Yeah, could be one of the parameters. Like, as you are rightly pointed out, definitely the kind of prey that is available in the region and other parameters, it could be temperature, other, all those things may actually contribute for venom variability. But actually, people have just studied the venom variability. They have collected the venom from snake species available in different geographical location, and they have just analyzed. But at the venom gland level, nobody has touched, because it comes under Wildlife Act. We are not supposed to kill and study venom gland and all. That is the bottleneck. Yes, you are, what you are saying rightly pointed out that. So venom variability, yes, the kind of prey that is involved, and the prey availability may be vary from region to region. The Rajasthan may be different, in Gujarat may be different, and Karnataka, Tamil Nadu may be different. They're accessible for different kinds of prey. So depending on the prey and also depending on the local environment, I think the profile, venom profile also varies. That needs definitely detailed study. That requires venom gland, removal of venom gland and study other parameters. That is not possible because it comes under Wildlife Act. Not supposed to touch. Yeah. Wonderful question. Yeah. Any other questions?
No,
sorry, I could not complete. We could not complete because of some technical glitch associated with the computer. This thing. It is not my problem. It is the computer problem, online problem. Yeah, yeah, yeah. Question, please. Yeah, if there are no questions, shall I say thanks?